Okay, so good morning everyone. This lecture is going to be about prediction of protein structure, in particular secondary structure. So first we need to define what is a secondary structure element. So it can be defined as a consecutive fragment of a protein sequence which corresponds to a local region in the associated protein structure, showing distinct geometric features. So in general, about 50% of the protein residues participate in alpha helices and beta strands, and the remaining half is more irregularly structured, let's say. So it does not mean that the rest is really completely disordered. It can adopt different types of secondary structures that are less canonical than helices and strands. So the problem we try to solve here is, given an input protein sequence, we would like to output the protein secondary structure. And the assumption that we're going to make is that amino acids display preferences for certain secondary structures. So the idea is that the secondary structure is encoded in the protein sequence, so meaning that different letters will lead to different secondary structure. So here you have just a schematized representation of secondary structure along the sequence. So you have the sequence in amino acids, and then you can visualize the helices and the strands. So what is the motivation for predicting secondary structures? Well, it can be useful for fold recognition. For example, it could confirm a structural and functional link between between two proteins where sequence identity is low. It can help in structural determination in conjunction, for example, with NMR data or as ab initio prediction first step. It can help in the refinement of sequence alignments, possibly as a first step to structure prediction. So the idea is that if you have this low sequence identity, then you're going to use the prediction of secondary structures, and you're going to try to align the structures that are the same, and this is going to improve your alignment, your correspondence between the two proteins. It can also be useful for classification of structural motifs and for protein design. So here again, this is like in the first lecture, you have these 20 natural amino acids that we know. So they're along the backbone. So you can see the backbone is just this vertical stretch, and you have the side chains that point to the left, to the right. And you have, well, the name of the amino acid and also the one letter code. So for example, for arginine it's R, for lysine it's K, etc. So you can see on the figure that the atoms are colored according to their charge or their polarity. So for example, the two first residues, the two first amino acids are arginine and lysine, and they contain positively charged atoms. So these are positively charged, in particular nitrogen atoms. Then you have the aspartate and glutamate, which are negatively charged. Then polar amino acid, asparagine, glutamine, etc., etc. For example, you have the sulfur in yellow here. And then you can see that at the bottom you have really these aliphatic chains, and sometimes aromatic, which are really hydrophobic. Okay, so then there is also another information here, is the color of the names, which correspond to the preference for secondary structures. So for example, you have amino acids that will prefer to be in alpha helix, some in a beta sheet, and some which are called structure breakers. So as in the first lecture, I showed you some classification of the amino acids according to their physicochemical properties, and I told you it's not the same, it's not a unique classification, so there are different classifications that exist. It's just one among several. So here it's a bit the same. These are tendencies, preferences, but we cannot really take them for granted. So you may find in different papers different types of preferences, different insights. So take this, I mean, with care, let's say. So how do we define these preferences? Well, there can be reasons that come from the physical chemical properties, right? So the volume, the total charge, the partial charges, the bipolar moments, things like this. And so just to remind you, the proteins are composed of a hydrophobic core, which is shelled from water by a hydrophilic surface. So there is always this interface with water, which has to be hydrophilic, and then the core is really hydrophobic. So typically we expect the helices and sheets to be compacted in the core and then loops to be exposed to the solvent or to interact with substrates. So what are methods to predict secondary structures? Here is a possible classification of these methods. You have, and you will see that in fact in a structure prediction we always have this kind of distinction between what we call empirical methods, which rely on, let's say, the knowledge that we have acquired on the objects we're looking at, so based on the physical chemical properties of the amino acids. You have statistical methods, which are more agnostic, maybe. They just use the information we have in structural databases to derive some statistics and then predict based on these statistics. Then you have machine learning method, which you can see as a kind of advanced version of statistical methods, if one can say that. So even more agnostic here, you think, okay, I'm not even gonna put some constraints on the way I derive the statistics. I'm just gonna use a machine that will learn from all the observed data. And you have hybrid or consensus methods, which are going to take advantage of the different methods that exist to make some kind of meta prediction. So right now this problem is essentially solved. We have about 80% accuracy for the best modern methods, even more. So there used to be weekly benchmarks for assessing the accuracy. I'm not sure it's quite the case now. But essentially, the predictions are very good, that achieve can be very high, and the delta that there still is with the observation can be explained by the fact that you cannot. There is a theoretical reason for which we cannot predict 100% of the secondary structures. So I will come back to this later. Okay, so just to give you a brief history of the field, so empirical methods are first, because at first we didn't have any observed data, so we didn't have any statistics, let's say. So in the 60s, there were a couple of papers, several papers that came out, and they were trying to see preferences based on the few structures that were solved at the time. So, for example, Guzzo in 65 observed that non-helical parts of proteins were mainly composed of proline, aspartate, glutamate, and histidine, and this was based on just two structures, that of hemoglobin and that of myoglobin. Then more structures came out, lysozyme, ribonuclease, etc. And this rule was formulated by Proteor that 5 consecutive amino acids are in a helix if at least 3 are alanine, valine, leucine, or glutamate. So then in 69, there was a formulation to form break helices. And then Lim came out with 14 rules in 74, so almost 10 years after, to predict alpha helices and beta sheets. Okay, so the idea really at the beginning was to try to infer some rules that we could write down and that could explain the preferences, the arrangements that were observed in the few structures we had. So again, if you have a 3D structure, it's direct to infer the secondary structure. So this is not prediction, right? This is just assignments. So you can rely on this gold standard secondary structure that you have in 3D structures, okay, to then test your prediction method, which starts from the sequence. So here is an example of an observation and a kind of rule which is interesting, I think, also because it proposes a representation of helices that is, yeah, interesting. So the idea is that you're going to take your helix and you're just going to project it on 2D. So it's just like the helix is here, and you look at it like this, and you project on 2D, right? And you just color, so you can see this as a network, and you just color the amino acids, which are represented by the nodes, according to their hydrophobicity, right? So that's what you have here. You have the yellow ones, which are non-polar, so essentially hydrophobic, and the green ones, which are polarly charged. What you can see is that you have a nice division between those that are hydrophobic and those that are polar. They're really on separate side of the helix. So there is one side which is contacting water, and one side that is protected or shelled from the water, okay? So there is this tendency to localize on one side. So another empirical method that is very interesting and important, I think, because it gives a lot of information, is the hydrophobic cluster analysis. So again, here we have a 2D projection of the secondary structure elements. So what's the idea? The idea is that you're just gonna, so you take the 1D sequence and you're just gonna put it on a helix, okay? And then you cut the helix in two and you unfold it. You unfold it, so you end up with what is shown in B. You have those diagonals, NAT, AIF, etc. Okay? And you duplicate this. So you have this unfolded helix duplicated. So you put it at the bottom as well. And when you duplicate, you slightly shift it. Okay? NAT and AT. So that's what you have in C. So when you do this, you have patterns that appear that correspond to secondary structure elements. So you can see this with the colors. For example, the sheet is going to be this kind of vertical strands of hydrophobic residues. So hydrophobic residues tend to cluster together when they're part of a secondary structure. For the helices, it's more something more horizontal that you can see. So you have two helices. And again, they correspond to, you're looking at hydrophobic residues, okay? It's a hydrophobic cluster analysis. So you identify those clusters, and based on the patterns, you can also identify the secondary structures. So here, how do you distinguish that there are two helices? Because the pattern probably changes. So historically, this was really manually done, so by visual inspection. So there is now an automated version of this tool, which is not, I think, published, but which is available on GitHub, so you can check. So it's a very powerful method, but yeah, for a long time it was really manual. Okay, so two examples here of common clusters. For example, you have an archetype of alpha helix, an archetype of beta strands. Okay, so what you can see here is that you can encode the hydrophobicity and the belonging, let's say, to the secondary structural element with 0 and 1s. And so again, you have the pattern that correspond to either the strand or the helix, and really the residues that allow you to identify those structures. So you can see that the pattern roughly correspond to the pattern you have in 2D in the secondary structure between the hydrophobic residues MMFL. Okay, it really looks like the pattern you have on the paper, and also this IWWV. So that's not all the residues, right, that allow you to identify the secondary structure element, because here you have really the PDBs, and you can see that there is more, but these clusters at least allow you to detect. So it doesn't mean a cluster is really the same length, represents the same length as the secondary structure element. Okay, so you can see these clusters as the building blocks of protein domains, right, because they represent secondary structures. So here you have typically an analysis of the cluster length according to the number of hydrophobic residues, and you have a very good correlation, right? So you can have on the diagonal, you see those that are perfectly correlated. And what you can see is that you don't have anything on the top left corner, so meaning that you cannot have some very small clusters with a lot of hydrophobic residues, but that's something expected. And then you can see that you have different distribution of these dots. Okay, so for example, you have we can localize here some particular proteins, particular types of proteins, protein domains. So for example, the globular protein domains are going to be at the bottom. So you can have a very varied length with not so many hydrophobic residues. And you will have also membrane things where you have membrane domains where you have a high length and also a high number of hydrophobic residues. So by looking at these relationships, you can identify different types of domains. So these are colored dots, and then for the white dots, you have something that is really too small. Okay, so very small clusters, very few amino acids, hydrophobic residues, you can't say much. And those are just too large. So meaning that you have, let's say, a length that can be high and a middle number of hydrophobic residues. So in between, it's not very clear. Okay, so again on this, there's been a recent study of providing a database of these hydrophobic clusters, so that's very interesting also to look at this to see what are the most common types of cluster you see. Also because of these binary encoding of the clusters is very convenient, and there are a few observations that you can make. So you have example here of some logo which show you what type of cluster, what type of sequences represent one type of cluster. Okay, so that's so I encourage you to read more about this. It's very interesting. So going back now to the prediction with statistical methods. So we've switched. There is nothing showing it, but we've switched from empirical to statistical. So it's roughly at the same time. You see the first one is 74. So it's a bit later than empirical because we need sequences, as I said before. So a basic naive idea for doing a statistical method is just to count occurrences of each of the 20 amino acids in each structural motif, right? So you're going to have the probability of having a motif C given the type S, which will be the number of residues of type S in motif C, divided by the number of residues of type S. Okay, and C is what? C is the three possible secondary structure you can have: alpha, beta, or gamma, which is all the rest. Okay, that's usually what we do, and then you can have final resolution. And what is the type then? S is just the amino acid type. Okay. So basically, you just count and you normalize by the number of by the frequencies of occurrence of the different amino acid type. So you classify the residues according to their propensities. So for example, here you have an example taken from the paper where you see that you have amino acid that will form helices, that will form sheets, some that will break them, and then you can refine the predictions based on a series of rules. That was the idea. Then a bit later, so that's about 4 years later, you have the Gore algorithm which came out, and it was really improved over the years, so it evolved. So here it's based on information theory combined with Bayesian statistics. And an important thing is that it accounts for the influence of the neighboring residues. Okay, before we just had single column independent statistics. Now we have a window and we can account for the neighboring residues. So the size of the window is typically 8. Minus 8 plus 8, sorry, so that's typically more like 16. It doesn't look like this here, but okay. And so you're gonna just make a computed product of the probabilities based on sequence counts. So it's the same formulation for the probability based on the counts, and you're just gonna multiply for the different position in the window, right? Okay, you normalize to avoid bias toward the most frequent structural motive. And then in the improvements, you have pairwise interactions of the neighboring residues that are accounted for. So here it's just really to account for the neighboring residues, but without explicit representation of the coupling of the dependencies you can have. It's just a product, so it's essentially independent. But then you also have pairwise interaction in improvements. So a very important thing is that these first methods were really improved by the use of multiple sequence alignments. Okay, so that's what you can see on the figure. It's not just about considering each sequence associated to its structure. It's each sequence and also similar sequences, which we assume have the same structure, because structure deviate much less than sequences, right? So the assumption is that proteins with similar sequences display similar secondary structures. And then you have a probabilistic representation of a sequence associated to structure, and this allows you to improve a lot the predictions. So machine learning method now, we can mention this neural network that was developed. There is no reference, by the way, so maybe that's just to give you an idea. So step one of the algorithm, it learns to recognize complex patterns. So for example, sequence secondary structure association in a training set. And then you determine the weights, and once the weights are fixed, the neural network is tested, right? So just training tests. So here you have basically one hidden layer. That's all. The input layer is the just the encoding of the protein sequence, and the output layer again is alpha, beta, or coil, gamma, if you wish. And so here in this implementation, you have sigmoidal function for integration. So this was, of course, before deep learning architecture really emerged. So it's basic, I would say, classical neural network architecture. And so the initial sequence is again defined by a sliding window, 10 to 17 residues. You have the 20 amino acid types that are considered and 3 as the output. So this is typically what is used in the PhD methods. So they used, so here the idea is that you have even multiple, so there is already a notion of piling up the layers, if you wish, because there are multiple neural networks that are trained and that produce predictions. So you go from sequence to structure for the first network, and then from structure to structure, and then you make the arithmetic average of both networks. That's the idea. Okay, so there is also Cypred, which uses this, and it was in fact much simpler. You still have 2 networks. So why is it much simpler? It's still input, hidden layer with 17 units, 75 units, and 60 for the other one, and the 3-state prediction. And you start from raw profile from PsyBlast. Okay, so then how do we evaluate the performance of these methods? So there is the Bi-Residue Score, basically the percentage of correctly predicted residues in each class. Okay, so Q3 will be the numbers of residues correctly predicted in alpha, beta, gamma, divided by the total number of residues to which secondary structure was assigned, and you put that in percentage, okay? So typically the data contain, let's say, 32% alpha, 21 beta, 47 gamma. So random prediction performance would lead to 37%. So if you're better than this, you're better than random. You can also assess this by segments instead of residues. So here you're going to compute a segment overlap. Okay, so you have the length of the actual overlap between the observed and predicted segments, and you have the length of the total extent, the union of both, right? So just intersection and the union, and you do the ratio between the two. And then you have some normalization coefficient. So the data, of course, must be separated in training and test set, but that's true for a lot of tasks. So here you need to be careful that there is no significant sequence identity between the training and the test set. You need also to have a representative test set to assess possible bias from the training set. And you need to define a gold standard. So you can do cross validations. So for the historic and most popular method, we have Shu and Fassman, which give 52. So this is the Q3. 52, Gore is 62, Gore 5 is 73.73.5, PhD is 73%. So the theoretical limit is estimated as 90%. Okay, so right now I think the best method are like 84, 85. And this 90% is why? Because some of the secondary structures are really stabilized by the tertiary structure, so they cannot really be predicted starting from the sequence. And there are also, protein can switch between different secondary structures. So this can be provoked or triggered by some interaction with the environment. And actually this is becoming more and more evidence that these things happen and they are not rare. So let's say you have a contraction of a protein at the part, and then the beta strand is going to become half a helix or something. Consensus methods. So these are obviously those that were the best. Okay, 82, 83, 85%, Shipred, Concord, Porter 5. So these are meta servers. They use a lot of tools, they compare them, and they just combine in consensus scheme. And so I put this Porter 5, which is very recent, 2019, so last year, over ensembles of cascaded bidirectional recurrent neural network and convolutional neural network models. So you see that, for example, here this deep learning architectures arrive at 84, which is not a dramatic improvement compared to what we had before. Okay, so that's exactly just to illustrate this this secondary structure switch when things are not that simple. So this is called extend fold switching. And you have examples in different homoligomers. So for example, you can see here you have the blue and the red, and the secondary structures are different, okay, and these are the same proteins. So they oligomerize, let's say, and once they are in oligomers, they change their secondary structure from the monomeric form. So you have the second example of click 1 also, where you can see that really you have first you have just two helices, and then you have something completely different. I mean, you have one helix, a small part of helix, and then strands, completely different. So this is probably to stabilize the interaction in the homoligomer. So there is also a study that is worth mentioning in 2016, large-scale assessment of order to disorder transition. So that's also something very important, is that sometimes we have intrinsically disordered proteins and they just become ordered by interacting with some partners. So here the question is, what are you going to predict based on the sequence, right? Because it's very much context dependent. Okay, so we'll stop here for now, and tertiary structure will follow.